As we begin this discussion, let's first consider a cell that undergoes division, goes through the cell cycle, and creates two new cells. Okay, so we know quite a bit about what happens with the nucleus and the chromosomes in the nucleus, how they divide and are separated during mitosis so that we get two nuclei, one in each of the new cells, and that they are identical. Now, in order for these cells to be equally functional, we can't ignore other things in the cell, namely organelles. So let's only talk about one organelle here, just for simplicity, but this would be true with other organelles as well. The mitochondrion, and let's just draw two here. But there are more mitochondria in one particular cell. Before cell division can be completed, we need to do a couple things in regards to the mitochondrion. We have to make twice the amounts of lipids to make the membrane structures of the mitochondrion, and we have to have 2 times the proteins found in these organelles, so that when these two cells form, new cells form, they each have a complement of mitochondrion. If you didn't increase the lipids or the proteins, it would be hard to imagine that you would get equal mitochondrion in the two new cells. And as I said before, this is not just mitochondria. We could throw the peroxisomes, the lysosomes, the ER, all of that in here, and all that also has to double so that our new cells are fully functional. So once we have a cell that's formed, either these two here or the one pre-existing one, there has to be a way to move proteins that are made in the cytosol to individual organelles. So let's talk about that next. So let's take on this discussion of how do proteins get to their organelle of function. That is, if you have a mitochondrial protein that's formed in the cytosol, how does it get to the mitochondrion? I'm going to describe 4 different ways. Some proteins are made in the organelle where they will function. So if we have our cell here with our nucleus right here, and we have straw chloroplast, remember the chloroplast and mitochondria have their own genomes. I usually just draw one, but remember, each one of these chloroplasts has many copies of their genome. For the chloroplast to do its job, it needs these genes. These genes will be transcribed and translated in the stroma of the chloroplast. So, some proteins, they don't have to be transported because they're already there. Now, there's going to be other proteins that need to be imported in here, and we'll talk about those in a moment. The second way is nuclear proteins will be transported through a nuclear pore. So in this cytosol here, we will have ribosomes. And these ribosomes will make proteins, and if they're a nuclear protein, they will go through what appears to be a little hole here called the nuclear pore. And I'm just going to put NP for space issues. But it's a little hole in the membrane of the nucleus that allows proteins to move through it. I should mention that it's a very selective process. Not any protein can get through here. Only proteins that are needed for nuclear function. The next way is unfolded proteins move into ER, mitochondria, chloroplast, and peroxisome through a protein translocator that's located on the membrane. So since I have the chloroplast written here, what happens is there is these transmembrane proteins that function to allow a protein that's not folded yet to go directly through this translocator. And once in here, then they'll fold properly. Using this mechanism here, a fully folded protein can't make it through. I didn't stress it, but with this second one here, this allows a fully folded protein to go into the nucleus. So let's go ahead and add that right here. And again, just like with the nucleus, it's very specific as to which proteins are allowed in. This fourth mechanism involves some proteins move to lysosomes, Golgi, and the plasma membrane using transport vesicles. So let me draw the ER here, and I'll draw the Golgi stacks here. In this process here, a protein that is made in the ER folded in the ER, will leave the ER in a small vesicle. And this small vesicle will contain that protein, that folded protein. This vesicle now will fuse with the Golgi. And then this vesicle will leave one part of the Golgi and go to the next. And at some point, it will break away from the Golgi, and it may go to the lysosome, it may go to the plasma membrane. It will find a new destination. But the key is that they use these transport vesicles that originate from ER. Now, as I have said before, it's very specific as to which proteins go to which organelle. How does it know? How do chloroplast proteins, how do mitochondrial proteins, how do peroxisome proteins know that they are peroxisome proteins or mitochondrial proteins? So they're able to reach their proper organelles because of very specific signal sequences on the protein. And some of these I'm going to mention in general ways, and others I'm going to be more specific about. So import to the ER. That requires a large amino acid sequence at the N-terminus of a protein. And when we talk more specifically about ER import, it'll make more sense, I think, why it has to be at the N-terminus. Next, let's talk about a sequence that is necessary for retention in the ER. These are important because there are some proteins that need to stay in the ER, and because of the nature of their function, sometimes they're associated with vesicles that temporarily leave the ER. But in order to get back, they have to have a very specific signal sequence at the C-terminus. And this sequence it's a short one, so you should remember this one. It's called lysine, aspartic acid, glutamic acid, and leucine. It's right at the C-terminus of it. It's often just referred to as the KDEL sequence for the single letter abbreviation of each of these amino acids. So often we just refer to this as a KDEL sequence. So mitochondrial import, like the ER, it also has an N-terminus signal. There's some variability on this actual signal, and it's a fairly large one, so I don't need you to remember the whole sequence, but you should know it's at the N-terminus. For nuclear signals, so remember, nuclear proteins, they are fully folded when they enter the nucleus. So they don't require the signal to be at any particular point. It just needs to be exposed on the outside of the protein. And so this is neither N-terminal or C-terminal, it's just anywhere on the protein. And it's a series of prolines, lysines, an arginine, a lysine, and a valine. So you can see a series of positive charges here, which are important in the process of moving a protein into the nucleus. So I forgot to mention, this is an import sequence to get the protein into the nucleus. We also have nuclear export signals. They can also be anywhere in the protein. And so that's what I'm going to put here. You don't need to remember this sequence, anywhere in protein. And the last one I want you to consider are peroxisome import signals. And I'd like you to memorize this one. This is a serine, lysine, leucine. And this doesn't have to be specifically at the N-terminus or C-terminus. I think often it's at the N-terminus, but it doesn't have to be.